It is 9 in the morning. The fast is over. You are standing in your kitchen, 14 or 20 hours without food, and you are hungry. And whatever you reach for in the next 60 seconds is going to determine whether the biological program your fast has been running continues into the hours ahead or collapses before you have finished eating. Most people at this moment reach for whatever is quick, familiar, and available. Toast, juice, a protein shake, a bowl of cereal. And they have no idea that several of those choices are sending a specific molecular signal that shuts off autophagy, reactivates mTOR, disrupts the gut microbiome consolidation the fast produced, and replaces the biological state they spent those hours building with its functional opposite. Not because of how much they eat, because of what they eat. And the mechanism by which each of the worst foods does this has never been explained in the specific molecular terms it deserves. Until now. Here is the biology that makes the first meal so critically important. When you fast, two master molecular switches are in the position that produces the benefits most people fast for. The first is AMPK, the cellular energy sensor that activates when cellular energy availability is low. AMPK activates autophagy, drives mitochondrial biogenesis, improves insulin sensitivity, and suppresses the inflammatory pathways that accelerate cellular aging. The second switch is mTOR, the mechanistic target of rapamycin, which is in its suppressed state during fasting. mTOR is the master regulator of cell growth and protein synthesis. When nutrients are abundant, mTOR is activated and it shuts off autophagy. When nutrients are scarce, mTOR is suppressed and autophagy runs. Think of AMPK and mTOR as opposite ends of a seesaw. When AMPK is up, mTOR is down. The fasted state holds the AMPK side elevated. The moment specific foods arrive, the nutrient-sensing machinery detects them and begins driving the seesaw in the opposite direction. Some foods drive it gently and slowly, allowing a gradual transition that preserves some of the fasted benefits. Others drive it violently and immediately, slamming the seesaw to the mTOR side in minutes and shutting off autophagy before the maintenance crew has finished its work. The GeroScience 2025 pilot randomized clinical trial on fasting mimicking diets and autophagic flux made this precise point from the opposite direction. Participants who broke their fast with the correct nutritional profile maintained elevated autophagic flux 2 days after refeeding began. Participants who broke with a different macronutrient composition lost the autophagy signal within hours of eating. The nutrient composition of the first meal after a fast determines whether the cellular repair program continues into the refeeding period or collapses the moment food arrives. And a separate review published in Science Direct in August 2025 confirmed explicitly that high glycemic obesogenic diets suppress autophagy and intensify oxidative and endoplasmic reticulum stress through mTOR reactivation. The worst foods are not simply unhealthy foods. They're the foods that most specifically and most completely send the molecular signal that terminates the biological program your fast was running. And these mechanisms scale with the duration of the fast. Breaking a 16-hour fast with toast is less consequential than breaking a 72-hour fast with the same toast, because the deeper the autophagy runs and the more completely the gut microbiome has been enriched, the more there is to lose in the first 10 minutes of eating. Here are the 6 categories. Category 1. High glycemic refined carbohydrates, bread, white rice, crackers, cereals, pastries. Breaking a fast with refined carbohydrates is the single most efficient way to immediately end everything the fast was producing. Here is exactly why. After a fast of 12 hours or longer, your insulin is at your overnight minimum. Your GLUT4 glucose transporters have been activated through the insulin-independent fasting pathway, and your muscle cells are highly sensitive to glucose. Your mTOR is suppressed, your autophagy is running, and then you eat toast, crackers, or a bowl of cereal. Refined carbohydrates are digested and absorbed almost immediately. Within 15 to 30 minutes of eating white bread or a high glycemic cereal, glucose floods your bloodstream at a rate that far exceeds what fasted skeletal muscle can absorb through the GLUT4 pathway alone. The pancreatic beta cells detect the surge and release insulin in a large first-phase response. Insulin binds to the insulin receptor. The PI3K and ACT signaling cascade activates. ACT phosphorylates and activates mTOR1. The mTOR seesaw slams to the opposite side. Autophagy is inhibited within minutes. The maintenance crew that was working inside your cells is sent home. The glucose spike itself produces a second problem specific to the post-fast state. After a fast, your cells have been running autophagic programs that have reduced oxidative waste inside them. The sudden glucose surge generates reactive oxygen species through the mitochondrial electron transport chain at a rate proportional to the substrate availability. The cellular environment that the fast had cleaned becomes rapidly repolluted by the oxidative exhaust of burning a large glucose load that arrived too quickly to be managed efficiently. And a third problem specifically for the gut microbiome. During the fasting period, the beneficial bacteria Akkermansia muciniphila and bifidobacterium have been selectively enriched through the removal of dietary substrates that feed less beneficial species. Research published in the Journal of Al-Qura University 2025 confirmed that intermittent fasting increases microbial diversity, particularly enriching bifidobacterium and Akkermansia muciniphila. The moment refined carbohydrates arrive, rapidly fermentable sugars feed the faster-growing opportunistic bacteria preferentially, beginning the reversal of the microbial enrichment the fast produced before the beneficial bacteria have had time to consolidate their position. Breaking a fast with refined carbohydrates is the nutritional equivalent of sending the maintenance crew home, turning the factory back on at full production, and flooding the newly cleaned floor with the waste product of the production process simultaneously. What to eat instead of refined carbohydrates? Cooked and cooled resistant starch, such as cold sweet potato or legumes, produces a significantly lower and slower insulin response than refined equivalents because the resistant starch fraction bypasses small intestinal digestion and reaches the colon as a prebiotic fiber rather than a glucose flood. Category 2. Fruit juice and sweetened drinks. Fruit juice is the most misleading category in this list because it is commonly recommended as a gentle way to break a fast. The biology says otherwise. Fruit juice delivers fructose and glucose in a liquid form that bypasses the satiety signals and gastric emptying delays of whole food. A glass of orange juice delivers approximately 25 grams of sugar, predominantly a mixture of fructose and sucrose with no fiber, no protein, and no fat to slow gastric emptying. Liquid sugar enters the small intestine within minutes of consumption. The absorption rate is among the fastest of any food form. The fructose in fruit juice goes directly to the liver through the portal vein, where it is metabolized through a pathway that bypasses the rate-limiting regulatory step of glycolysis. As described in the cholesterol and triglyceride scripts in this series, fructose arrives at the liver's production floor without going through inspection. The liver has no mechanism to limit how much fructose-derived carbon enters de novo lipogenesis. All of it gets converted to fat. In the post-fast liver, which has depleted its glycogen stores and is operating with maximum fat oxidation capacity, the arrival of fructose from juice directly competes with the fat oxidation program and begins redirecting the hepatic machinery toward fat synthesis instead. The glucose component of fruit juice simultaneously drives the insulin spike, the mTOR reactivation, and the autophagy termination described in Category 1. But the fructose adds the additional hepatic mTOR activation and de novo lipogenesis burden that whole fruit, with its fiber-mediated slower absorption and lower fructose delivery rate, does not produce to the same degree. Breaking a fast with fruit juice delivers the maximum possible glucose-fructose dual spike through the fastest possible absorption pathway, terminates autophagy through simultaneous hepatic and peripheral mTOR activation, and begins reversing the microbiome enrichment and fat oxidation program the fast established. What to drink instead of fruit juice? Water with a pinch of mineral salt and a squeeze of fresh lemon provides electrolytes and the trace citric acid that supports mitochondrial function without delivering the fructose-glucose dual mTOR spike that juice produces. Category 3. Ultra-processed foods. Ultra-processed foods, the packaged products with multiple industrial ingredients, including emulsifiers, artificial sweeteners, refined oils, and flavor enhancers, represent a specific category of fast-breaking mistake that goes beyond the glycemic problem of refined carbohydrates. The emulsifiers commonly found in ultra-processed foods, including polysorbate 80 and carboxymethylcellulose, directly disrupt the gut mucus layer that protects the intestinal epithelium. Research published in Nature confirmed that these specific emulsifiers alter the gut microbiome composition and increase intestinal permeability, the so-called leaky gut that allows bacterial endotoxins to cross the intestinal barrier and trigger systemic inflammation. The fasted gut, with its Akkermansia-enriched mucus layer and reduced intestinal permeability from the fasting period, is particularly vulnerable to this disruption at the moment of first refeeding. The first food to arrive at the intestinal epithelium after the fast is the food that interacts with the gut lining that has been maintained and consolidated by the fasting period. Ultra-processed food emulsifiers begin degrading that consolidated lining before the first meal is finished. The artificial sweeteners in ultra-processed foods create a second specific problem. Research including the 2022 Nature Medicine study by Suez and colleagues confirmed that specific non-caloric sweeteners, including saccharin and sucralose, alter gut microbiome composition in ways that impair glucose tolerance. In the post-fast state, where insulin sensitivity is at its best and the microbiome has been enriched by the fasting period, immediately exposing the gut to artificial sweeteners that impair glucose tolerance and microbiome diversity is a direct negation of two of the primary benefits of the fast itself. What to eat instead of ultra-processed foods? Any single ingredient whole food with no added industrial components, an egg, an avocado, a piece of cooked fish. The gut lining that the fast consolidated interacts with these without emulsifier degradation or artificial sweetener microbiome disruption. Category 4. Dairy-heavy meals and high-protein first meals. Protein and mTOR have a specific relationship that most fasting content has not addressed directly enough. mTOR is activated by two independent signals. The first is insulin from glucose. The second is amino acids, particularly the branched-chain amino acids leucine, isoleucine, and valine. Leucine is the most potent known activator of mTORC1 through the regulator-RAG complex. This creates a genuine tension in the fasting community that needs honest framing. You want to eat protein at your breaking meal to preserve muscle mass and stimulate muscle protein synthesis, and protein directly activates mTOR and suppresses autophagy through the amino acid sensing pathway regardless of insulin. This does not mean protein is the enemy of fasting. It means timing matters. A very large protein load, particularly from dairy, which is rich in leucine, as the very first food after a fast, delivers the maximum possible amino acid-mediated mTOR spike at the moment when mTOR is most completely suppressed and autophagy is most deeply running. The practical implication is that a smaller amount of protein consumed after an initial gentle reintroduction of fiber and fat allows the transition from the fasted state to the fed state to occur more gradually, with less abrupt mTOR activation than a large dairy-heavy meal as the first post-fast food. What to eat instead of a large dairy-heavy first meal? 2 to 3 whole eggs provide complete protein with fat from the yolk that slows gastric emptying and moderates the amino acid delivery rate to mTOR, producing a more gradual mTOR activation than whey protein or a large serving of Greek yogurt as the opening food. Category 5. High inflammatory processed oils. Processed seed oils rich in omega-6 linoleic acid, including sunflower, corn, soybean, and canola oil, drive arachidonic acid production and the prostaglandin and leukotriene inflammatory cascade that the fasting period has been suppressing. During fasting, the production of anti-inflammatory eicosanoids and the reduction of pro-inflammatory signaling through NF-kappa B suppression is one of the mechanisms driving the reduction in systemic inflammatory markers that regular fasting produces. Breaking a fast with foods fried in or heavily processed with omega-6 rich seed oils delivers the substrate for renewed arachidonic acid production directly into a metabolic environment that has been moving in the opposite direction. The prostaglandin inflammatory cascade reactivates. The NF-kappa B suppression that fasting was maintaining begins to erode, and the endothelial protection from the fasting period is challenged by the lipid peroxidation products that oxidized polyunsaturated fats produce when heated. What to use instead of processed seed oils? Extra virgin olive oil or avocado provide predominantly monounsaturated oleic acid, which does not feed the arachidonic acid inflammatory cascade, and both provide the fat that slows gastric emptying and moderates the transition from fasted to fed without the oxidized polyunsaturated fat burden. Category 6. Alcohol. Alcohol at any point undermines the fasting biology, but breaking a fast with alcohol is a specific compounding error. The liver, which has been running its optimal fat oxidation and ketone production during the fast, is diverted immediately to alcohol metabolism when ethanol arrives. The liver prioritizes alcohol clearance over all other metabolic functions through the alcohol dehydrogenase and aldehyde dehydrogenase pathways. Fat oxidation stops. Ketone production stops. The gluconeogenesis that was maintaining stable blood glucose is disrupted. The autophagy that was running in hepatic tissue is inhibited through both the mTOR reactivation from the caloric content and the direct suppression of autophagy by acetaldehyde, the first metabolite of alcohol metabolism. Research published in the journal Autophagy confirmed that ethanol and its metabolite acetaldehyde inhibit autophagic flux in hepatocytes through direct suppression of lysosomal membrane integrity and LAMP2 expression. The liver's cellular maintenance program is shut off at the organelle level by the specific chemistry of alcohol metabolism. What to have instead if alcohol is planned for the occasion? Consume it later in the eating window rather than as the fast-breaking first calorie. Even a 1 to 2 hour delay after breaking the fast with food allows the liver to transition partially from the fasted metabolic program before alcohol metabolism competes with it. The post-fast liver is the most vulnerable to alcohol-mediated autophagy suppression precisely because its autophagic machinery is running most actively. Here is the honest framing. The GeroScience 2025 pilot RCT on fasting mimicking diets and autophagic flux is published and peer-reviewed. The Science Direct August 2025 confirmation that high glycemic obesogenic diets suppress autophagy through mTOR reactivation is published. The Journal of Al-Qura University 2025 review on mTOR and autophagy dynamics is published. The Nature research on emulsifiers is published. The 2022 Nature Medicine research by Suez and colleagues on artificial sweeteners is published. The Autophagy journal research on acetaldehyde and lysosomal membrane suppression in hepatocytes is published. The fructose and hepatic de novo lipogenesis mechanism is established across the Journal of Clinical Investigation. The leucine and mTOR1 regulator RAG activation is established across decades of mTOR biology research. The honest caveat is this: the autophagy suppression from refeeding is not uniformly bad. The fed state and mTOR activation are required for muscle protein synthesis, immune cell function, tissue repair, and dozens of other biological processes that the fasting state deliberately suppresses. The goal of breaking a fast correctly is not to avoid mTOR activation indefinitely. It is to allow the transition from the fasted to the fed state to occur gradually, with the most biologically compatible foods arriving first, rather than slamming the molecular seesaw from one extreme to the other with the most disruptive possible first meal. If you have been doing any of the above without knowing the mechanism, your fasting results have not been canceled, they have been reduced. Every fast still produces benefit. The question is how much of that benefit survives the first meal.
Here is what to eat to open the first meal after a fast. Bone broth or mineral water with electrolytes first. A small amount of avocado or olive oil to slow gastric emptying without an insulin spike. Cooked, non-starchy vegetables to provide prebiotic fiber for the microbiome. Then 2 to 3 eggs or a small portion of fish after 15 to 20 minutes. The complete transition should occur across 30 to 60 minutes rather than in a single large meal that slams every molecular switch simultaneously. Here is the complete picture. Your fast has been running AMPK up and mTOR down. The autophagy maintenance crew has been working inside your cells. The gut microbiome has been consolidating the beneficial bacteria the fast enriched. The fat oxidation program has been running in the liver. The inflammatory environment has been quietening. The foods in the 6 categories above each slam the mTOR seesaw in the opposite direction with varying degrees of violence and through different mechanisms. Refined carbohydrates slam it through insulin-mediated mTOR1 activation. Fruit juice adds simultaneous hepatic fructose-driven mTOR activation. Ultra-processed foods disrupt the gut lining and microbiome that the fast consolidated. Large, dairy-heavy protein loads activate mTOR through the regulator RAG-leucine sensing pathway. Inflammatory seed oils reignite the prostaglandin cascade the fast was suppressing. And alcohol shuts off the liver's fasting metabolic program through the acetaldehyde-mediated autophagy inhibition in hepatic tissue. The fast works. The refeeding determines how much of that work survives into the hours and days that follow. Drop a comment right now. Which of these 6 categories surprised you most, and has this video changed what you will eat for your next post-fast meal? I read every single one. If this permanently changed how you think about the first meal after your fast and why what you eat at that moment determines how much of the fast you actually keep, hit subscribe. Every week on the Rewired, we decode the mechanisms behind the habits that determine your health, your brain, and how you age. New mechanism every week. You spent the hours building the biological state you wanted. The first meal decides whether you walk out of the fast or whether you slam the door on your way in.